Hello, good evening. Good evening, everyone. Is my voice is clear? बताओ कमेंट में बताओ. Voice clear है ना? Good evening, Vijayali. Ranjan, good evening. Meena, Madhu Egde, Rimi, good evening. Yeah. Yes, okay. Yeah, Kavita, good evening. Sir, please bring this session in daytime. अरे भाई डे टाइम तो मतलब सारे लोग अवेलेबल नहीं रहते हैं ना. रात में 9 बजे 10 बजे तो सब लोग आराम से रिवाइज कर सकते, मतलब एक बार रिवीजन ऐसे हो जाता है ना. डे टाइम कुछ लोग कॉलेज में जाता है, कुछ लोग दूसरा प्रिपरेशन करते रहते हैं. बताओ कमेंट में बताओ किसको ये सेशन से ना सारे चल रहे हैं ना वो रात में चाहिए, किसको मतलब डे में चाहिए, देखेंगे. मतलब बताओ कमेंट में बताओ, डे टाइम और नाइट टाइम. बताओ ना. So this is at night time. Night is best time. Night. देखो देखो सारे सबको नाइट ही बेस्ट है. ठीक है, मतलब कुछ लोग को डे टाइम. गुड इवनिंग सिंचन. गुड इवनिंग अंकुर, आदित्य. शेयर कर दो फटाफट, सबको बताओ. आज हम डिस्कस करेंगे 2024 पेपर. ठीक है, ये तो एक बार डिस्कस ऑलरेडी हुआ था, मैं फिर से एक बार कर रहा हूँ. हेलो हेलो, वॉइस तो क्लियर है ना? मतलब इज इट लो और ओके? एक बार बताओ. भी ಬ್ರಾಡ್ಕಾಸ್ಟ್ ಮಾಡಕ್ಕೆ ಹೇಳೋ. सो यस सर, क्लियर है. ठीक है. सो इन प्लांट बिडिंग अनडिजायरेबल लिंकेज कैन बी ब्रोकन बाय रिकरेंट सिलेक्शन, डिस्ट्रप्टिव मेटिंग, डेली सिलेक्टिव, ऑल ऑफ द अबव. रत्नेश ग्रुप में भी लगाया था आप ऑलरेडी. फिर इतना जल्दी इधर भी लगाया है क्या? मैं अभी देखा था वो क्वेश्चन. अभिषेक सर इज द बेस्ट मेंटर फॉर ब्रीडिंग. अरे संजू चौधरी, ठीक है. बताओ अभी बताओ कौन सा बेस्ट है, मतलब the breeding method which by using that we can break the undesirable linkage. ऑप्शन में रिकरेंट सिलेक्शन है, डिसरप्टिव मेटिंग भी है, डायलर सिलेक्टिव मेटिंग भी है. बताओ आंसर बताओ. ऑल, सिंचन बता रहा है ऑल. ठीक है. दूसरा लोग, सर मैं कंफ्यूज्ड वो ऑप्शन बी में. ठीक है, ऑल. यस, you can just say रत्नेश. ठीक है, undesirable linkages we can break by intermating. Okay, we can break by all possible number of crosses like intermating. Okay, you can see in diallel selective mating what we will do, we will do all possible matings in F1 or F2 plants, okay, among themselves in diallel selective mating. In recurrent selection also, using testers after the selection of after the evolution of progenies, which are the possible best phenotypes which are they are giving, we will select the seeds first, then we will do intermating among themselves. And in case of disruptive mating, name itself suggests, okay, we will try to cross between two different phenotypical in two different extreme phenotypes. In case of disruptive mating, we will select only the extreme phenotypes there. So your variability are increasing there. Also you can break the linkage. Okay, in case of disruptive mating also. Answer to all it clear, Ratnesh. In all of the cases, we will try to break the undesirable linkage. Okay. ओके ठीक है 41 शेयर कर दो फटाफट सबको शेयर कर दो चालू करेंगे ओके 50 चला गया आज फटाफट ओके शुरू करेंगे अभी फर्स्ट क्वेश्चन विच ऑफ द फॉलोइंग ऑर्गेनाइजेशन ठीक है primary responsible for the collection evaluation conservation of plant genetic resources in India with for research purpose okay B ठीक है आंसर तो NBPGR okay which comes under your Indian Council of Agriculture Research located in New Delhi. If I ask you the question who is the responsible for origin of NBPGR, okay, who is the main fellow behind the establishment of NBPGR, पता है किसी को? We will also call him as if I yes Remy, he is Harbhajan Singh. We will also call him as Indian Vavillo. You can see the National Bureau of Plant Genetic Resources. It is first started as Plant Introduction Scheme in 1946. Okay, under the Division of Botany in IARI. Okay, in IARI. Okay, under Division of Botany, it started as Plant Introduction Scheme in 1946. Then in 1956, it converted as Plant Introduction and Exploration Organization. It converted as an organization again under the Division of Botany. Now there is no Division of Botany in IARI, but in the initial years. Then in 1961, it separated as Division of Plant Introduction within IARI. Okay, in IARI, as case of your state agricultural universities, okay, we are not having departments. Okay, we are having divisions. There is a 26 divisions in case of IARI, like genetics and plant breeding, seed science and technology, division of pathology, division of entomology. Okay, like that. First, it's under the Division of Botany. Then it is separated as Division of Plant Introduction as a separate division in 1961. In 1976, okay, separate bureau, okay, National Bureau of Plant Introduction in 1976. In 1977, it renamed as NBPGR, National Bureau of Plant Genetic Resources. ठीक है, ये तो हिस्ट्री है NBPGR का. It started with Plant Introduction Scheme. Then in 1977, it actually got a name, National Bureau of Plant Genetic Resources. Love of India, Vavillo of India. Remy, ठीक है. The National Bureau of Plant Genetic Resources, there are different objectives. Okay, germplasm management, plant quarantine, crop improvement. Okay, the National Gene Bank, one of the main objective of NBPGR. Okay, India's first National Gene Bank established in 1996. Okay, under NBPGR. It also having National Herbarium for cultivated crops. It is having total 10 regional stations in different agroclimatic zones. Okay, the important thing is the National Gene Bank, okay, which is established in 1996. ये तो पता है आपको, ठीक है. सब लोग ऑल इंडिया टूर आते हैं ना, इसी के सामने फोटो लेके जाता है ना आप लोग. The primary objectives of conservation of the plant genetic resources are the primary objectives. They are asking for choose the correct answer from the option given below. बताओ कौन सा निकालना है इधर से. B. Answer is B. ADE only. You can see that to conserve genetic diversity for future generation, one of the main objective. to promote monoculture farming practices. अरे भाई हम जर्मप्लाज्म कंजर्वेशन कर रहा है, इसलिए हम डाइवर्स जर्मप्लाज्म को कलेक्ट करने के लिए, ठीक है. द मेन ऑब्जेक्टिव इज टू रिजर्व और टू रिस्टोर द डाइवर्स जर्मप्लाज्म इन द एग्रीकल्चर, ठीक है. वो मोनोकल्चर क्यों जाना है? ओके, इट विल नॉट प्रमोट द मोनोकल्चर ऑब्वियसली. टू एराडिकेट इन्वेसिव प्लांट स्पीसीज, it is also not objective of germplasm collection. To ensure food security and sustainable agriculture, yes. To facilitate research and innovation in plant breeding. If we are having germplasm resources, that means we are having variation. Variation is the, okay, in plant breeding, variation is starting point for each and everything. So it will do that one, okay. So your answer is A, D, E only. Option is B. Select the correct statement about origin of the tetraploid and hexaploid wheat. Okay, the tetraploid species, that is Triticum turgidum, is an allopolyploid. It is right. A, B, right? Your Triticum turgidum. The hexaploid species, Triticum aestivum, is an allopolyploid. Again, it is right. A, B, D. Triticum turgidum arose from combination of Triticum monococcum and Triticum timpovi. सही है क्या? आप लोग तो आंसर लगा रहा है. It is not correct. Triticum aestivum arose from combination of Triticum turgidum and Aegilops toschi. This is the right answer. You can see, if I want to write evolution, it will start with Triticum urartu into Aegilops speltoides. Cytoplast 1. Cytoplast 1. Cytoplasm 1. Okay. You will get Triticum turgidum. In Triticum turgidum, there will be so many subspecies. Your durum, dicoccum. Okay. Those are subspecies. This Triticum turgidum again will get crossed with your Aegilops toschi. Okay, this is A, B, A, B will cross with D. You will get Triticum aestivum subspecies aestivum. This is the evolution of your wheat crop. If they ask the donor parent of each genome for Triticum aestivum, okay, you should choose Triticum urartu, not monococcum. Okay, monococcum also contains a genome, but it will another lineage. Okay, for Triticum zukowski. Okay, there is another lineage. Okay, for the Triticum zukowski. This is also hexaploid wheat. Okay, for this a genome donor is your Triticum monococcum. For Triticum aestivum, okay, Triticum aestivum, your A genome donor is Triticum urartu. B from Aegilops speltoides, cytoplasm 1. Then your D genome from Aegilops toschi. ठीक है, clear? बताओ. Yeah. Okay. Given below are two statements. Okay. In nature, the tetraploid and hexaploid wheat reproduce as diploid. Okay. Reason, in the presence of PH1 allele, chromosome 5b, in chromosome 5b, each chromosome pairs only with its homologs from same genome and reproduces as a diploid. ठीक है, आंसर बताओ. ये your both A and R true and R is correct explanation of A. You can see here. For this condition, okay, the pairing only between the diploid genome. Okay, we are having some term. कोई बताओ, कमेंट में बताओ. For this present condition, okay, you can see pairs with only its homologue from the same genome and they will reproduce as diploid. Amphi-diploid, yes, good. This is the condition of amphi-diploid. In case of wheat, okay, we are having AA, BB, DD genome. They will not pair among themselves. Okay, they will only pair as AA, BB and DD. When there is a meiosis, okay, they will pair and they will separate. Okay, as AA, BB, DD. They will not pair among themselves and they will not give meiotic irregularity. Okay, why? Due to this pH allele. Okay, when there is in dominant condition, they will pair as normal AA, BB, DD. So they will again behave as diploid. But when there is a mutation in pH locus, okay, when there is a mutation, there is a possibility of homologous pairing. Means they will pair among themselves. Okay, when we want to transfer some wild gene, okay, when we want to transfer from any gene from another wild parent of wheat, okay, we can't directly cross our Triticum meastium with that particular another genome. Okay, there will be so many genomes. Okay, apart from AA, BB, DD, there will be a Z, there will be a R genome if you want to take it from rye. In that case, we will use pH1 locus, that is mutated pH1, so that there will be pairing among other genomes also. So we can easily transfer from the wild genome. Okay, homologous pairing, yes, Rimi. It will promote homologous pairing, your pH1 locus. Next statement. Amplified fragment length polymorphism, that is your AFLP, is a type of molecular marker that relies on enzymatic digestion, yes, and PCR amplification. ये तो क्लास में बताया था ऑलरेडी में. Statement 2. Your RFLP is a type of molecular marker that detects variation in length of DNA fragment generated by digestion with restriction enzymes. Both statements 1 and 2 are true. In case of RFLP, what I have told, this is your genome. Okay, you will use restriction enzymes to digest your DNA, so it will cut into different length. So after this, what you will do, you will use the probes. Okay, you will use the probes and your probe will attach here. In this particular case, another genome having cut like this, your probe, you will use same probe, your probe will attach here. So you can see it is 5 kb, this is 10 kb. You can see the variation in case of RFLP. Okay, but in case of AFLP, what you will do? First, this is your genome. First, you will use again restriction enzymes. It will cut into different fragments. Then you will use primers. Okay, the selective primers. It will only amplify this kind of products. Okay, both restriction enzyme and PCR. In this particular case, AFLP, but here only restriction enzyme digestion. ठीक है, ये तो ऑलरेडी मैं एक्सप्लेन किया था क्लास में, समझ रहा है ना. अभी बताओ इसमें कोडोमिनेंट कौन सा है, डोमिनेंट कौन सा है, बताओ कमेंट में लगा. Which one of these marker is dominant and codominant? Sir, I didn't understand. अभी मैं मार्कर क्लास नहीं ले सकता इधर, ठीक है, मतलब सारे एक्सप्लेन नहीं कर सकते, मैराथन में करेंगे ये अभी 2/2. ठीक है, अभी बस इस समय आप समझना चाहिए क्या RFLP बेसिक प्रिंसिपल क्या है RFLP का. ठीक है, दिस इज जीनोमिक DNA in the both the cases. Just we will use restriction digestion here. We will just use restriction digestion and will using by probes we will see the fragment size difference. Okay, the basic principle is fragment size difference after the restriction digestion. But in this case, in case of AFLP, okay, we'll use both restriction digestion, then after that PCR. Okay, we'll not use probe here, we'll just use primers and we will selectively amplify some of the products. So in some plants we will get presence, in some plants we will not get that band absence. Here presence absence. ठीक है, सब लोग लगा रहे हैं RFLP को dominant. Yes, good good. AFLP dominant. ठीक है, ये तो एक्सप्लेन किया था. टोमेटो समझ गया ना? अभी इतना याद रखना. ठीक है, मैराथन में I will try to cover. Which of the following organisms naturally possess CRISPR-Cas9 system? ये भी मैं क्लास में बताया ऑलरेडी, सारे एक्सप्लेन किया है पूरा. ठीक है, how it will evolved, how it got evolved with CRISPR-Cas9, then how we are using it. रिमी अंकुर क्लास तो सुना? श्रुति मतलब क्लास तो लिया है ना? ये मैं अपलोड किया था शायद आपका कोर्स में, मतलब पीएचडी के लिए किया था, वो बाद में पीजी स्टूडेंट्स को भी अपलोड किया था. देखा है क्या आप लोग? डिड यू गाइस सी इन दिस क्लास इन आवर कोर्स? रिमी, श्रुति, अंकुर, आप लोग, कविता, कॉन्जुगेशन एफ प्राइम फैक्टर. अरे नूतन क्या लगा रहा है तू? ये क्यों आ रहा है बीच में? नो सर. सिद्धार्थ नो सर. जाके देखो एक बार. श्रुति यस सर, श्रुति सिंह यस सर, और रिमी यस सर. ठीक है, इसमें दिस इज द immune system, natural immune system in bacteria. Okay, you can see here. I will explain, don't worry. CRISPR is nothing but clustered regularly interspaced short palindromic repeats, full form. Okay, how we discovered it? Okay, this is the natural phenomenon, your CRISPR. Then we modified it and now we are using to target genome editing. Okay, प्लांट्स, अरे प्लांट्स नहीं है भाई, अग्री प्रैक्टिस. ठीक है, इधर देखो, इट इज योर बैक्टीरियोफेज, नथिंग बट योर वायरस. ओके, बैक्टीरियोफेज नथिंग बट योर वायरस. द वायरस व्हिच अटैक्स बैक्टीरिया इज बैक्टीरियोफेज. ओके, what it will do? It will inject its DNA, viral DNA, into bacteria. So bacteria, what it will do? It will cut into different pieces. Okay, and some of that part, okay, some of the part of viral DNA, it will insert it into its own genomic DNA. This is the bacterial DNA. It contains Cas operon and tracer RNA gene and the spacers. These spacers are nothing but, okay, your viral DNA. Whenever your virus, that is bacteriophage, attacks the bacteria, okay, the sum of the part of your viral DNA will insert into this CRISPR RNA. You can see this is the one of the bacteriophage DNA. This is also bacteriophage DNA. This is also bacteriophage DNA. Another bacteriophage. Like this, whenever some bacteriophage attacks, it will cut and it will store some information from that bacteriophage. Okay, then it will produce its RNA. It is DNA, then it will produce RNA. We will call that as a CRISPR RNA. Okay, then this Cas operon, it will produce Cas9. Cas9 is nothing but protein molecule. Okay, it is like molecular scissor. Okay, it is endonuclease. Okay, like it will cut. It is the objective or the function of that Cas9 enzyme will cut the DNA. Okay, then you can see the same viral bacteriophage, the virus attacks, if attacks again, you can see this is the viral DNA. Okay, it is already stored in bacterial genome. It will produce that a counterpart, that is CRISPR RNA. This CRISPR RNA, okay, and this tracer RNA, it will give stability to that CRISPR RNA and the Cas9. These will come together and it will bind to this particular viral DNA. Since your CRISPR RNA is complementary to this particular DNA, since it is already inserted into your bacterial genome, so you can see This is viral DNA. Okay, viral DNA. This is your CRISPR DNA. It is produced from the spacers. Okay, it complementary. In that particular situation, your Cas9 will come. Your Cas9 will come. This is your Cas9. It will cut the viral DNA. So this is natural adaptive immune mechanism for bacteria identified in 1987. Clear? Sir, please speak a little in Hindi also. ठीक है, समझ गया ना? Simply for you guys, what you should know, okay, it is natural adaptive immune mechanism for bacteria. Okay, it is naturally occurred in bacteria, so we have modified it for our purpose. What will happen? Bacteria phase रहता है ना, वो अटैक करता है बैक्टीरिया को, वो कुछ डीएनए है ना, उसमें कुछ पार्ट स्पेसर के अंदर जाता है, इधर स्टोर करता है बैक्टीरिया स्पेसर के अंदर, ठीक है. When the same bacteria phase infects again, ठीक है, उस समय इसका कॉपी बनता है ना, वो इस डीएनए का एमआरएनए कॉपी बनता है, इधर डीएनए आता है, इसका कॉम्प्लीमेंट्री पार्ट इधर से आता है, ठीक है, वो बाइंड हो जाता है, योर Cas9 प्रोटीन रहता है ना, वो आके उसको कट करता है, ठीक है. नेक्स्ट इफ इट अटैक्स अगेन, then there is no infection to the bacteria. Okay, that's why we will call it as adaptive immune system. ठीक है. In eukaryote, Cas9 used in year 2013. Yes, now the recent scientists, they are working on humans also. Okay, genome editing in humans. Now they are using your Cas9 in eukaryotes also. सिंचन, ठीक है, समझ गया ना? मतलब बेसिक तो समझ गया ना CRISPR का. This is basic idea. So now we are using this CRISPR RNA. We will we will try to target in plants. Okay, we know the information, sequence information. We will try to build this CRISPR RNA. Okay, we will try to design this CRISPR RNA as our modification, so as our target. So we'll use this CRISPR RNA, and we will also use Cas9, and we will cut in the plants wherever we need to cut. So there will be a mechanism, SDN1, SDN3, SDN1, SDN2, SDN3. So you can insert, you can delete, or you can also insert the new complete gene. Okay, this is the basic idea. GM paddy variety also released. Agree practice. वो GM paddy variety नहीं है भाई, वो जीनोम-एडिटेड राइस है, ठीक है. कहाँ से आपका नाम क्या है? अग्री प्रैक्टिस 2026. GM पैडी वैरायटी नहीं है, ठीक है. GM पैडी रिलीज नहीं हुआ है इंडिया में, वो जीनोम-एडिटेड राइस है, ठीक है, नॉन GMO, ठीक है. Which of the following is not serious limitation for conserving the plant genetic resources under in vitro repositories? They are asking about not a serious limitation. You can see all other three options are limitations. Genetic stability may be compromised over time due to accumulation of somoclonal variation. Yes. Okay, under in vitro condition, there is a possibility of somoclonal variation. High cost associated with the establishment and maintenance of in vitro collection. Yes, it is also limitation. Vulnerability to catastrophic events such as power outage and equipment failure. Yes, this is also limitation. Limited capacity to conserve large collection compared to field gene bank. This is not a limitation. Okay, there will be a more capacity to conserve large collection in case of in vitro condition. Okay, you can go up till 10,000, 20,000 of collection, but in field gene bank it is very difficult to conserve. Okay, your answer is C. Good. इधर देखो, अभी मैच द फॉलोइंग. इसको तो मैं कुछ नहीं बता रहा है अभी, ठीक है, आपको ऑलरेडी पता है. हम लिस्ट भी दे दिया है, कोर्स में भी दे दिया, मैं प्रीवियस क्लास में भी ऑलरेडी दे दिया है. किस किस क्रॉप में कौन सा फाइटोकेमिकल है, एंटी न्यूट्रिशनल फैक्टर है, ठीक है. सिंचन तो लगा रहा है DD, ठीक है, A3 erucic acid brassica, your trypsin inhibitor is glycine max, BOAA in lathras, phytic acid in Ziamase. And in classes I have already covered, okay, how we can decrease erucic acid, how we can inhibit your trypsin inhibitor, okay, how we can improve your folic acid. There will be so many genes, okay. This is the basic idea. Definitely they will ask one question on this. Which of the following are important varieties of sugarcane? Where there is a co, that is sugarcane. Okay, no doubt. But here they are confused with one of the option. That is your current 9 and LRA 5166. कौन सा है इसमें? कौन सा वैरायटी सुगरकेन का नहीं है? आप एक निकालना है इधर से. ABDE आंसर लगा रहा है क्या? Current 9 is sugarcane. From where? Can anyone know from which institute the current 9? Leaf cotton. This is your cotton variety, no doubt about it. Okay, cotton variety. What about current 9? From which institute? They have the co are from SBI, Sugar Cane Breeding Institute, Coimbatore. What about current 9? This is also regional station of SBI. किसी को पता है क्या कहाँ है रीजनल स्टेशन ऑफ SBI शुगर केन ब्रीडिंग इंस्टीट्यूट? PAU नहीं नहीं नहीं, PAU में नहीं है. बताओ, कोई बताओ. तमिल, अरे वो तो तमिलनाडु में your SBI main institute. I'm asking about regional station of your SBI. Current 9 is from that particular regional station. Good, good Ankit. Good, good. Colonel. ठीक है. There is an institute, ICR IAWR, okay, Indian Institute of Wheat and Barley Research. Beside that particular institute, okay, in Karnal there is a regional station of Sugarcane Breeding Institute. Okay, that is in Karnal, Haryana. Yes, Vijayali. Good. Self-incompatibility, male sterility and pistillate condition have one effect in common, that is prevent self-pollination. Yes, it is right. Self-incompatibility and male sterility are used in hybrid seed production, while pistillate condition is used in inbred development. Reason? बताओ. Co 05011, another name of current 9. ठीक है, ये तो मुझे पता नहीं था. D सिंचन T लगा रहा है. D, A is false but R is true. Great, Shinchu, you are great. भाई सारे D क्यों लगा रहे हैं? A is false. किसको आंसर दे रहा है आप लोग? मुझे कंफ्यूज कर रहा है. Self incompatibility, male sterility and pistillate condition have one effect in common, they prevent self pollination. Yes, they prevent self pollination. सिंचन क्या हुआ आपको? हाँ, अंकुर, सिद्धार्थ, सारे डी लगा दिया. रोजी, रिमी, आप भी डी लगा दिया. A is true and your R is false ना. Self in your pistillate condition also used for hybrid production, okay, not inbred development. Okay, it will also promote cross pollination, right? C, it was D by mistake. Okay, Shinchu, C will happen. C pistillate condition used in castor. Yes, वो तो पता है. ठीक है, ठीक है, अभी ठीक है. सब लोग पिस्टिलेट के बारे में बता रहा है. बताओ, कोई बताओ कि वो पिस्टिलेट कंडीशन में देयर विल बी 3 टाइप्स. ओके, there will be some term for pistillate condition in castor. What is that particular term? NES type. Yes, Kavita. You can see in pistillate condition, okay, in castor, N type, S type, and NES type. N type is nothing but your GMS. Okay, that is you are controlled by the nuclear genes. Your NN, small NN will male sterile. These are fertile. When you cross with small n and small n, you will get capital N, small n, small n, small n in 1:1 ratio against 50% sterile, 50% fertile. Okay, same as your GMS system. Your N type, S type, that is sex reversal variants. Okay, there will be a primary branches, secondary branches in case of castors. Okay, your primary branches will bear, okay, only pistillate flowers. That means only female flowers. Those are male sterile. Then that secondary branch and tertiary branch, they will start to bear the monoecious flower, monoecious inflorescence. Okay, both female and male. Okay, this is your S type. Okay, your primary branches will bear male sterile plants. Those are nothing but your pistillate, only female flowers. Okay, male sterile, your primary branches. Then after secondary tertiary branches, they will produce monoecious. In case of NES type, okay, less than 35 degree, more than 35 degree. Okay, you are less than 35 degree and more than 35 degree. Less than 35 degree will produce male sterility. More than 35 degree Celsius, they are monoecious. Okay, that means these are fertile. You will get both male and female flowers like TGMS and PGMS. It will controlled by temperature. Okay, clear? कुछ डाउट है क्या किसी को NES टाइप में? N टाइप, S टाइप and your NES टाइप, बताओ. क्लियर है ना? श्रुति, सिंचन, कविता, नूतन, yeah, yes, clear. Biology made easy. Biology made easy. ठीक है, हम बना रहे हैं प्लांट ब्रीडिंग मेड इजी. पता नहीं किसको, कितना लोगों को इजी हो रहा है, कितना लोगों को इस क्लास सुनने के बाद मुश्किल हो रहा है, मुझे पता नहीं. ठीक है, दिस इज आवर कोर्स. Okay, now you are getting at 1,999 for your exam, upcoming exam 2026. Okay, for next year your mock test bundle will be 799 and your course will be for 4,999. And for 2028 exam, okay, we are already launched that course also. You are getting at 8,999. Okay, you just, the one who are already purchased this course, you can just suggest your juniors. Okay, they will definitely get benefit from these classes. And one who are not yet registered, okay, you can just ask your seniors or anyone already purchased this course. You can take feedback, then only you just enroll to our courses, so that you will get an idea, okay, how to prepare and how we are giving the quality content to you guys. ठीक है, easy sir, easy बना रहा है क्या? ठीक है. Which of the following statements truly describes self incompatibility? ये देखो, यही पेपर में सारे सेल्फ इनकम्पैटिबिलिटी एंड मेल स्टेरिलिटी कवर कर दिया. ठीक है, ऐसे एक एक पेपर में एक एक कॉन्सेप्ट उठाते हैं. ठीक है, वो लोग. Self incompatibility prevents self fertilization in plants. Okay, they are asking about correct answer. Self incompatibility promotes outcrossing, thus increases genetic diversity within the plant population. Yes, right. Self incompatibility is a mechanism that allows plants to self fertilize effectively. They are already told that it will prevent the self fertilization. Okay, now they are again giving it promotes. Self incompatibility is controlled by genetic mechanism within the plant. Yes, it will be controlled by, yes, locus. Okay, okay, there will be so many number of genes. Sometimes it may be diallelic, it may be different number of genes. Okay, polygenes also control sometime from S locus. Okay, this is genetic mechanism. कौन सा आंसर है? डी 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 ए बी डी. ठीक है, दिस इज नॉट एन करेक्ट आंसर. आपको पता है ना सेल्फ इनकम्पैटिबिलिटी एंड मेल स्टेरिलिटी. In self incompatibility, okay, you will get functional pollens. Your pollens are functional, but they will not able to fertilize on the same plant. Okay, there will be a incompatibility reaction. If it falls on the same plant, there will be a reaction, gametophytic and sporophytic. But in case of male sterility, okay, your pollens only not functional. Okay, your pollens are not functional. But here in self incompatibility, there will be a functional pollens. Okay, but if they falls on same stigma of the same plant, then there will be a reaction. Okay, you should remember this particular basic principle. ठीक है? Yes sir, yes, same genotype. Cytoplasmic male sterility is commonly used in hybrid seed production in the following crops. They are asking, you should see commonly used in. We can use CMS in each and every crop, but they are telling that commonly used in hybrid seed production in the following crops. C, 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 A, B, C. अरे भाई A, B, C. आप कारण छोड़ रहे हैं, सब लोग C लगा रहे हैं. अरे भाई A, B, C, D, A, B, C, D. फिर से रंग. They are asking about commonly used. अरे भाई wheat में कहाँ यूज कर रहे हैं हम लोग? wheat में हाइब्रिड ही नहीं है पहली चीज. In wheat there are no hybrids. If they are available also, we are not using CMS. We are doing through ant pollination and ant emasculation. ठीक है? There is some hybrids, they are using chemical hybridization agents for that. कहाँ घूम गए सारे लोग? ये लगा रहे क्या? BCD. Good. In sorghum we know combined Kafir 60A. In sunflower, your pet one from Elianthus petiolaris. In case of your corn, your CMS T. Yes. Okay. In wheat we are not commonly using your CMS, and in your chickpea also. Okay. You should know that. Okay, if you are using CMS, then there should be a availability for pollination through insects or through wind. Okay, in case of sunflower, in case of sorghum, there might be a pollination through wind. In sunflower, there might be a pollination through wind and your insects. In corn also, there will be a pollination from wind again. ठीक है? चिकपी में कहाँ से होगा? विंड से. चिकपी फ्लावर देखा है ना आप लोग? Do you guys saw chickpea flower? There will be a minute flowers and also clistogamous. There will be a keel petal, standard petal. Okay, they will only opens after pollination. It is strictly self pollination crop. वो कहाँ से आप क्रॉसिंग कर सकते हैं? ये CMS यूज करके. If you want to do the hybrid in case of chickpea, then you should go for ant emasculation and ant pollination. ठीक है? CMS in vegetatively propagate crops commonly used, sir. Yes, we can use keel petal pressy. ठीक है? ये तो concept है. ठीक है? Where there is an availability for pollination through wind or insect, there only you can use CMS, so that there will be natural crossing. If it is from ant pollination and ant emasculation, then why you will go for CMS? Okay, it's not a possible outcome. Male sterility cytoplasm in crop plants usually arise from interspecific hybridization, self-incompatibility, spontaneous mutation, and induced mutation through ethidium bromide. Chemical hybridizing agents used in wheat. अरे भाई चिकपी में मुझे पता नहीं चिकपी में नहीं यूज करता है, ठीक है, बस वीट में यूज किया था, अभी भी वो उतना फेमस नहीं हुआ है, मतलब उतना सक्सेसफुल नहीं हुआ है, ठीक है, चिकपी में केमिकल हाइब्रिडाइजेशन नहीं यूज करते, ठीक है. That's why in chickpea the most of the crop varieties released as a varieties, okay, not through heterosis breeding, not through hybrid breeding, ठीक है. C A C D, yes, A C D. You can see male sterility cytoplasm through spontaneous mutation. Those are your CMST, CMSS in maize. Those are spontaneous mutation. Okay, induced mutation through ethidium bromide. There are so many CMS sources. Okay, we have induced through ETBR. Okay, through ethidium bromide. Under specific hybridization. Okay, we can transfer the cytoplasmic sources from wild relative. Okay, from wild relative we can transfer the cytoplasmic sources. From Elianthus petiolaris, okay, we have transferred it. In case of Brassica, Ogura, okay, those are different cytoplasms. We have transferred from interspecific hybridization to our cultivable species. But so it is irrelevant. Okay, our self-incompatibility is very irrelevant to this particular question. Okay, A, C, D, A, C, D. Heterosis refers to the phenomenon where the hybrid offspring exhibits superior trait performance compared to the parents. Okay, this is the basic definition of your heterosis. Heterosis is primarily attributed to the combination of favorable alleles from each parent resulting in enhanced trait performance of the hybrid offspring. बताओ आंसर. Triticum tymphovi. उधर से हम in wheat we have transferred some cytoplasmic sources from Triticum tymphovi, Triticum caudatum. Okay, there are so many varieties, so many germplasms. From that we can through interspecific hybridization we can transfer the CMS sources. Yeah, yeah. Superiority over parents. Okay, the F1 superiority over parents is nothing but your heterosis. Okay, it is primarily attributed to the combination of favorable alleles. Okay, it is combination of favorable alleles from the each parent. Okay, that will enhance the performance of offspring. That is due to heterosis. Your both statements are right. Okay. Again आ गया आपका सिंथेटिक वैरायटीज, ले लो, चार मार्क्स आराम से ले लो इधर से, ठीक है. या डोमिनेंस इफेक्ट, यस सिंचन, योर heterosis is due to dominance. In 2025 paper, again they have targeted your heterosis breeding. Okay, we will discuss what exactly dominance, over dominance, different genetic basis, okay, in the next class. Synthetic variety, ठीक है. First one is collection of diverse inbred lines, then evaluation of inbred lines through GCA, okay, then crossing, then multilocation. Okay, simple चीज already explain किया था हम already, but each and every steps also I have already explained in the previous classes. D A C A D B, C A D B, answer is D, ठीक है. Next question, which of the following statements about synthetic varieties are correct? अरे क्यों दे रहा है इतना ऐसे क्वेश्चंस बार बार, ठीक है, आप लोगों के लिए इजीली 3-4 क्वेश्चन तो इजीली दे रहा है वो लोग, ठीक है. सिंथेटिक वैरायटीज आर हायर यील्डिंग देन हाइब्रिड्स, ये गलत बात है ना, ठीक है. सिंथेटिक वैरायटीज आर हेटेरोजाइगस हेटेरोजेनियस फर्स्ट ऑफ ऑल, and you are mixing the seeds from so many hybrids, there will be not that much yield as compared to your uniform hybrids. ABC, good. ठीक है, ठीक है, आ गया, इधर ले लो आपका मार्कर्स, प्रिंसिपल and the DNA marker. Okay, if you attended my classes, then you will easily answer this particular question. अरे भाई, किस किस को मार्कर्स के बारे में पता नहीं है ना, कोई दिक्कत नहीं, मैराथन क्लास में एक आइडिया दे देंगे, ठीक है। बट अगेन आई एम टेलिंग, it's very difficult to cover from the scratch in the marathon classes also. Okay, in class itself, okay, you can ask our students, it took 2.5 hours or 3 hours for marker class. Okay, it's very difficult to explain in the each and everything, but I will give some glimpse. Okay, the basic principles. See, ठीक है। Restriction digestion followed by PCR amplification of selected fragments. पिछले साइड में भी बताया था। First we will do the restriction digestion. We will cut. Then we will use some selected primers, selective nucleotides, and we will do the selective PCR. That is in your AFLP. इसमें नाम है, ठीक है, AFLP, amplified fragment length polymorphism. For A4, use of PCR to amplify repeated motifs of DNA. That is your SSRs. Okay, simple sequence repeat. Variation in single nucleotide within DNA. Those are your SNPs. Detection of variation in length of DNA fragments produced by restriction enzyme. That is your basic RFLP. ठीक है, आंसर तो आप लगा रहे हैं सी, ठीक है, मैं भी लगा देंगे, आप इतना कॉन्फिडेंसली बता रहे हैं, मैं ऑप्शन भी नहीं दिखा, आई एम नॉट सेइंग। ओके, दिस इज द बेसिक आइडिया, ठीक है। यू कैन सी RFLP इज कोडोमिनेंट। ओके, इट विल डिफरेंशिएट योर डोमिनेंट होमोजाइगोट एंड हेटेरोजाइगस। SNP अगेन कोडोमिनेंट, ठीक है। योर SSR अगेन कोडोमिनेंट, your AFLP dominant. Okay, your RAPD also dominant. ठीक है, important है, note it down. These are about your markers. Kavita Patil, multi-allelic. Yes, your SSR are multi-allelic. Multi-allelic nothing but, you know, if you are having some number of more than 2 forms of alleles for particular gene, you will call as a multiple alleles. Same like that. Okay, if you see in SSR, you can have more number of alleles. Some will give 80 base pair repeats, some are 100 base pair repeat, 120 base pair, 300 base pair, 400 base pair. Particular locus, you can see there will be a more number of repeats. Okay, based on the number of repeats, you will get an allele. So it is multi-allelic, your SSR. ठीक है, क्लियर? बताओ, क्लियर है ना मार्कर्स के बारे में, डोमिनेंस एंड कोडोमिनेंस नेचर? आप लोगों को उतना नॉलेज होना चाहिए, ठीक है, डोमिनेंट और कोडोमिनेंट एंड बेसिक प्रिंसिपल, ठीक है। टुबुटू आपका नाम क्या है? टुबुटू नाम क्या है आपका? तो लोकस स्पेसिफिक मतलब you know from which locus your allele is coming. Okay, you are targeting particular locus. In case of RFLP, it is locus specific. You are using restriction digestion and your probe. Okay, they will go and attach that particular locus only. So you are seeing the band and you know from where it is. Okay, in case of SNP, again you know the sequence information, it is locus specific. Your SSR, again locus specific. Okay, you know the loci from where exactly your SSR marker is present. But in case of RAPD and AFLP, these are not locus specific. We don't know from where exactly your DNA is amplifying. Okay, we will use random primer. That's why those are non locus specific. Sir, please sir, no name. ठीक है. SSR or high genotyping throughput. Okay, yes, good. Your SNPR now very high throughput compared to SSR. Okay. Which term is commonly used to describe the specific sequence of bases recognized by restriction enzyme? Which is the term we will use? Okay, we will use recognition sequence or restriction site. Okay, for each and every enzyme, okay, your restriction enzyme, there will be a particular recognition sequence. Okay, we will call that as a restriction site or recognition sequence. Yes, good. Nutan, Madhu, Shinch, Rimi, Kavita, good. See, okay. Objective of IT PG RFA. Okay. For food and agriculture. Again, it is very important. Signed in 2003, came into existence in 2004. It will promote conservation and sustainable use of plant genetic resources for food and agriculture. Yes. To ensure the fair and equitable sharing of benefits. Yes. These are the particular two objectives of your ITPG RFA. To encourage genetic modification of croplands and monoculture. It will not encourage any genetic modification and monoculture. Why it will promote monoculture? Okay, when we are conserving genetic resources, then why will go for monoculture? Yes, सिंचन. आप अच्छे से कमेंट डाल रहे हैं, अच्छे से इमोजी यूज कर रहे हैं. हाँ, आप लोग तो जेनजी है इसलिए. Facilitate access to plant genetic resources for food and agriculture. Good, exactly, especially for countries with limited resources and capacities. Okay, this is also one of the objective of your ITPG RFA. Strengthen the capacity of farmers, particularly those in developing countries, to conserve, manage and sustainable use of your plant genetic resources. Answer C. A, B, D, E only. Yes, your C is not an objective of your ITPG RFA. India is also member country in ITPG RFA. ठीक है? Years 2003 and 4 you should remember. अरे इतना ही आज. क्वेश्चन तो बहुत सिंपल था. ठीक है? And this paper they have targeted your markers, more markers and somewhat related to your self incompatibility and male sterility. Like this, in every paper they are targeting some of the parts, but in each and every paper your synthetics and composites, your anti-nutritional factors, in modes of reproduction they will ask, and your ITPG, RFR, PPFR, CBD, Nagoya, Cartagena, these are very common. Okay. Any doubts in this particular class? Do it in English. I am explaining in English only, right? बीच में इसमें थोड़ा हिंदी आ रहा है, मतलब हिंदी लोग भी है इधर, उसको भी समझना है ना. कुछ लोग बता रहा है सर हिंदी में बताओ, you just use some हिंदी. कुछ लोग बता रहा है इंग्लिश में कर दो. ठीक है, मैं किस में करना है? टू थैंक्स फॉर द क्लास. ठीक है, we will discuss the recent paper, that is 2025, very important. Okay, in nowhere you will get that particular discussion of that particular paper. Okay, we were doing it. Okay, we just attend tomorrow's session. Okay, which is very important. We will see what are the questions they have asked about in 2025 paper. Okay, English ये totally है, English ये totally है. सिंचन में English I am covering in English only, बस कुछ हिंदी यूज कर रहा है. You are our sunshine, नूतन. Thank you so much then. Okay, lighthouse of plant breeding. Arey bye. Okay, we will see in the tomorrow's class. Till then, tata, good night, bye. Yes, atrocious is in 2025 paper. We'll see what exactly atrocious is. Till then, take care. Thank you, Remy. Thank you so much. Thank you, Shinchan. Good night.